Polyphenols are micronutrients in our diet that are an emerging interest in the scientific field due to their abundance in our diet, role in preventing diseases, and role as antioxidants. There are hundreds of types of polyphenols, and in order to understand which types are the most beneficial to us, it is important to understand how they are broken down and distributed in the body. Not all polyphenols are absorbed equally. It is dependent on bioavailability, which is the proportion of a drug or substance entering circulation in the body that is able to have an active effect. There are several thousand molecules that have a polyphenol structure, which are classified into different groups based on the number of phenyl rings they contain and the structural element binding the rings. The four major groups of polyphenols are flavonoids, phenolic acids, stilbenes, and lignans. Flavonoids consist of two aromatic rings, bound together by 3 carbon atoms that form an oxygenated heterocycle. Flavonoids are split up by the heterocycle into 6 subclasses, consisting of flavonols, flavones, isoflavones, flavanones, anthocyanins, and flavanols. Flavonols are the most abundant flavonoid in foods, and the compounds are found in glycolated forms. Flavones are much less common than flavanols, and they consist mainly of compounds formed from simple sugars and another compound, either luteolin or apigenin. Luteolin is a flavone with a yellow crystalline appearance, while apigenin is a flavone found in plants. Isoflavones are flavonoids with a structure similar to that of estrogen. They contain a hydroxyl group configuration that corresponds to those of estrogens, which is why the two are very similar and also why isoflavones have the ability to bond to estrogen receptors. Anthocyanins are a type of flavonoid found as a pigment in plants. Depending on pH, they may be colored or uncolored. Flavanols are a type of flavonoid that are found in the monomer form as catechins and the polymer form as proanthocyanidins. Unlike the other flavonoids, flavanols are not glycosylated in foods. The second major polyphenol group, phenolic acid, can be split into 2 groups, one being a derivative of benzoic acid and the other being a derivative of cinnamic acid. Hydroxybenzoic acids are components of complex structures like hydrolyzable tannins. Hydrocinamic acid, on the other hand, is more common than hydrobenzoic acid and consists of p-coumaric, caffeic, ferulic, and cynapic acid. The most abundant phenolic acid is ferulic acid, which is an abundant photochemical found in plant cell walls. The third polyphenol group, lignans, are made up of 2 phenylpropane units. When lignans are metabolized in the intestine by bacteria, they form enterodiol and enterolactone. Stilbenes are the last group, and they are the smallest polyphenol group found in the human diet. They may have anti-carcinogenic effects, but are not consumed in quantities large enough to have productive effects. Polyphenols are found often as complex mixtures in a large variety of foods, and the abundance of them depends on factors such as the type of food, how the food is stored, and how the food is prepared. Fruit and beverages like tea or wine are the most abundant sources of polyphenols in our diet. Polyphenols such as quercetin are found in plant products like fruit, vegetables, cereals, legumes, fruit juices, tea, and wine. Different classes of polyphenols can be found in different foods. For example, galotannins found in phenolic acids are in mangoes, and the elegotannins found in phenolic acids are in strawberries, raspberries, blackberries, and other red fruits. Blueberries, kiwis, plums, cherries, and apples contain the hydrocinamic acid found in phenolic acids. Cereal grains contain ferulic acid in the outer layer part of the grain, including wheat, rice flour, and oat flour. Onions, curly kale, leeks, broccoli, and blueberries contain rich sources of flavanols, while celery and parsley contain mainly flavones. Isoflavones are found in high concentrations in soy products like soy milk, soybeans, miso, and tempeh. The catechins found in flavanols are present in some fruits such as apricots and very prominent in green tea and chocolate. The concentration of polyphenols found in these foods can vary depending on soil type, sun exposure, rainfall, and field or greenhouse culture. The concentration of polyphenols in a plant may increase as the size of the plant increases or if the plant is diseased, but it will decrease as the plant ripens. The way the food is prepared will also have an effect on polyphenol concentration. Simply peeling a fruit or vegetable can eliminate a majority of the polyphenols since large concentrations of polyphenols lie in the skin. The same goes for dehulling legume seeds or bolting cereals. Cooking can decrease polyphenol concentrations as well. The longer some vegetables are cooked, the lower the concentration of polyphenols will be. So, although potatoes have polyphenols present, by the time they are cooked into French fries, they have been stripped of all their polyphenols. Sorry, French fry lovers. Manufactured fruit juices have been stripped of many of their polyphenols as well because of how they are processed. The amount of polyphenols in the human diet can vary by region. Citrus fruits, which are the sole sources of flavonoids, are consumed majorly in areas where citrus fruits are most abundant. In Spain, apples, pears, grapes, and red wine provide large concentrations of catechins and proanthocyanidins. In Asia, isoflavones are consumed in much higher concentrations than they are in America or Europe because of the amount of soy products consumed in Asia. Tea, chocolate, apples, and pears aid in the large consumption of flavonols in Holland. The amount of hydroxycinnamic acids consumed can vary globally depending on the amount of coffee consumed. However, the most common polyphenols in our diet may not necessarily be the most active or beneficial to the body. Sometimes certain polyphenols are poorly absorbed, rapidly eliminated, highly metabolized, or have a lower activity in the body, even if they are the most abundant in our diet. In order for the health effects of polyphenols to be understood, it is essential that we obtain extensive knowledge of their bioavailability. Most cannot be absorbed in their native forms, and therefore must be hydrolyzed by intestinal enzymes first. The polyphenols are then bound to other substances which are more easily absorbed, allowing them to penetrate tissues in which they can be metabolized. The main protein responsible for binding is albumin, and the affinity polyphenols have for it varies depending on structure. Glycosylation has an increased effect on the absorption of polyphenols in the stomach, but their role and their interactions with acids there remain unknown. The amount of polyphenols found in the blood after ingestion vary according to the nature of the polyphenol and the food source. When green tea, red wine, and cocoa are ingested, the catechin and epicatechin are as effectively absorbed as quercetin. Anthocyanins have the lowest concentrations found in the blood after ingestion, while isoflavones are the best absorbed flavonoids. It is difficult to tell whether or not polyphenols accumulate in specific organs, but some studies indicate that some cells may readily incorporate polyphenols by specific mechanisms. Although the breakdown of polyphenols in the body is not yet fully understood, it is highly likely that they provide many health benefits. They have been found to have strong antioxidant properties by neutralizing free radicals and donating an electron or hydrogen atom. By donating a hydrogen atom to a free radical, polyphenols are able to act as a chain-breaking antioxidant. Polyphenols exert most of their antioxidant properties in the gastrointestinal tract, and the number of hydroxyl groups plays a factor in antioxidant activity. They may also act as co-antioxidants and aid in the regeneration of vitamins in the body. Polyphenols can have health benefits in animals such as rats, pigs, cows, and dogs. Pomegranate peels have been determined to have anti-inflammatory effects when added to cattle feed, and red wine has been found to have anti-carcinogenic effects, preventing tumors in animal and cell trials. This may display potential for breast cancer treatments. Research has also found correlations between polyphenol consumption and chronological diseases. Some studies have found that consumption of polyphenols can decrease the risk of high blood pressure, artery diseases, and high mortality rates. Studies have also shown that polyphenols can change lipid and energy metabolism, reducing risk of obesity and facilitating weight loss. Likewise, they have also been shown to reduce the progression of type 2 diabetes. Polyphenols are also likely to prevent and alleviate symptoms of metabolic syndrome, including hyperglycemia, dyslipidemia, and elevated blood pressure. The potential for polyphenols and their application in medicine in the future is promising, but will require extensive research to fully understand and learn all the benefits, effects, and possibilities of polyphenols.